All right, welcome everyone. It's my pleasure to welcome you all this evening. My name is Parvati Krishnan. I am the Director of Advocacy Alliance at Global Genes. It's my pleasure to welcome you all to the first pre-event webinar. Of course, our event is coming up on June 8th through 10th. It's our Rare Drug Development Symposium. It's taking place in Philadelphia. And we hope that you all will join us. And as a precursor to that event, we wanted to have these webinars that are leading up to it. So we hope to see you there. The link is in the chat, so please sign up. And what is unique about this event is it's going to be live and interactive workshop. It's not a hybrid event. It is an in-person event only. The 4 topics for the workshops are going to be listed here. They're disruptive innovations in clinical trials, AI-driven screening platforms, emerging models, and fostering successful collaborations and connections between stakeholders to accelerate progress. Again, we hope to see you all there in person. It's our first live event this year. And so we're looking forward to meeting you all. We have some amazing speakers. We have a lot of information that's going to be shared, and we would love to hear from you and learn from you about the work that you are doing. Without further ado, we have our first webinar here today, for which all of you are here. I'd like to introduce Karsten. Karsten is currently the Director of Translational Biology at Travere Therapeutics. He's a passionate scientist with almost 2 decades of research experience. When he embarked on his career path, he was motivated by scientific curiosity and the desire to help people who are suffering from diseases. This continues to be Karsten's driving force. It inspires him to work hard and to take challenges that may seem insurmountable. In the past years working in biotech, Karsten has been fortunate enough to witness how novel therapies can impact the lives of patients. Karsten feels that this is his calling, in particular in rare diseases that are neglected and have no available therapies. To find treatments for these diseases, he believes we need to have all stakeholders involved. Karsten is excited to work with patient groups, academic and governmental institutions, and specialized research laboratories to discover and develop new ways of treating patients. Karsten firmly believes that together we will make a difference. Over to you, Karsten.
A little bit nervous because this is the first time that I'm presenting on this particular topic. And I've made a few assumptions in preparing this presentation. And I apologize if some of this is too basic for some of you. And I want to encourage you to interrupt me if there's certain concepts that I'm rushing through in the process of presenting. I'm bound to fail to mention certain concepts or aspects. And I'd like to also encourage you to contribute and either put it in the chat box or at the end bring them up in our Q&A session. I realize that I'm not going to be comprehensive in covering all the topics that fall under this umbrella term. So yeah, I do believe this is a learning forum, and I do believe that I'm not the only teacher here. So please chime in where you feel like you want to contribute. So let's start with my disclaimer slide. I think we have to get through this at all times. I am an employee and also stockholder of Travere Therapeutics, and what I'm talking about today is really a reflection of my opinions and views, not necessarily those of my employer. I think the second disclaimer here is that a lot of what I've learned about this topic, I've learned from patient advocacy groups that we work with, so our partners in patient advocacy, and so I'm indebted to your community in teaching me about this topic as well. Okay, my introductory slide. I mean, this is some of my career path. I don't think it really matters that much, but I do want to spend like maybe a minute or two talking about myself, my motivation. Although Bhagavathi has also already mentioned that. But even when I was a child, my best friend's dad used to call me professor. And I don't think that was necessarily a compliment. He was sort of a businessman, but I would never cease to recite facts that I had read in books. In fact, that was my passion, and I would always lecture and keep lecturing. And so you could say that I was predestined to become a scientist. But then there was another side of me that really had a propensity to want to help people. And for example, when we were in a group setting, I would always approach the kid that was left out and would hang with them to make them feel included. And I'm fortunate enough to be in a position today to be able to marry those two passions of mine, being a scientist and being able to really interact with the patients that I serve, that I try to find treatments for, to see the impact of my work. And I'm indebted to to my current employer for giving me that opportunity. And along these lines, I also want to thank you for taking the time out of your schedule, because it is another way that I feel I can contribute to your community in trying to help you guys, become better organizations. And hopefully, I'll be able to do that today and give each one of you some snippets of information that help you move forward. The 2 points that I want to talk about on this slide are the scientific lead on Alagille syndrome project. So we have a Alagille syndrome project that we are conducting in collaboration with the Alagille Syndrome Alliance. And we talk about this topic a lot. We discuss it on a regular basis. So again, these are some of the people that I've gleaned insights from. I also work closely with our business development group, and we look at a lot of different disease areas. And I've developed maybe an eye for what makes a disease area promising from a drug developer perspective, versus one where maybe there's a lot of work to be done. And so I think I'm trying to sort of combine both of these perspectives in order to give you some advice today, point you towards sort of helpful resources, et cetera, et cetera. All right, quick introduction of Travere Therapeutics. So we are headquartered in San Diego. We're a company of approximately 300 people, and we're centered on the patient in rare disease. So really our values are all centered around rare disease patients, and we try to help them in any way that we can. We have a firm belief that we need to bring all the stakeholders to the table in order to solve some of the problems that we're facing in rare disease research and drug discovery. And so that's why we like to work with different entities, and Parvati mentioned some of that already. Now, our pipeline spans really from some commercial products all the way to really early discovery programs. In the middle of it, we have multiple different clinical assets in the pipeline. I'm not necessarily going to walk you through this pipeline, but I do want to mention the Alagille program and the NGLI1 collaboration that we have on here. Both of those are in collaboration with the respective advocacy groups. So we're working with CDG Care on the NGLI1 deficiency program, and with Alagille Syndrome Alliance on the Alagille program, and with the sort of a drug discovery branch of the NIH called NCATS. And we really believe that as a very successful model, and it's the right way of going about drug, sort of some even as early as discovery stage programs. So, see, here we go. As I was thinking about what I want to spend time on today, there were several themes that kept reoccurring. And I listed them here, and this is really sort of the overview of today. I don't know whether we will get to all of these. It's possible that we won't get to the last one. I do have slides on this, so I don't know whether you make the slides available, but if they're available, then you can take a look at them. Hopefully they're somewhat self-explanatory, and they will help you with learning some of the language that we use. So ultimately, the aim, of course, is that we understand each other, even though we have very different backgrounds, and some of us have very formal training, while you guys sort of thrown into the deep end. We often don't respect that, and we don't really recognize it enough. All right, so the first topic I really want to touch on is your research network. And I included physicians here. Some of them are physician researchers. Sometimes clinical research is something that you would consider. But from my perspective, being a translational biologist, I'm mostly going to focus on sort of preclinical research that is really sort of the understanding of the disease. And then we're going to move on to registries and natural history studies. That is something that we value very highly as drug developers. It helps us understand the disease better, and how it demonstrates itself in different people and so we can glean a lot of information from these studies. Third is really a tool, which are the biobanks and repositories. I'm going to try to point you towards a few different resources that will help you develop a biobank, if you don't have that already. And then I talked about 4 already. It's really just about jargon. So I have a question for you to start us off, and maybe out it out. Hopefully that's going to work. But my question here is, what do you think are some of the reasons why you want to expand your research network or tighten that network?
Well, this is Tamia Austin with the As1 Foundation. Our mission is sickle cell education with an emphasis on sickle cell trait literacy. Why I feel that expanding our research is critical is because there is still a lot that's not known about sickle cell trait, and the general knowledge that's spread is incomplete, which is the consideration that sickle cell trait is benign and asymptomatic. There's also a rare kidney cancer associated with sickle cell trait called renal medullary carcinoma. And doctors, physicians are undereducated about all of it. So we need more research, and every research article, more research is needed. So.
Yeah, thank you, Tamia. Anybody else?
We have a lot of feedback coming up in the chat as well.
Okay, I don't have the chat box.
I can read it for you. To get perspectives to address and solve unmet needs with unique sustainable methods, to expand network because we need some out-of-the-box thinkers. Not every nail needs a hammer, and to be more inclusive, to overshadowing in DUP 15Q is detrimental to patients and families.
Very good.
Thank you for adding in the chat.
Very good. Okay, I really like all of those. I think incidentally, they're not quite the same as I have, which is probably not surprising because we come from a very different angle. But I think that's exactly where we meet in the middle. And I want to give you my perspective. This is really from a researcher's point of view, and how it can enable research. And ultimately, of course, the goals are the ones that you mentioned, which is really education. We educate physicians, we educate families, and we educate the general public about the severity of disease, about the prognosis, et cetera, et cetera. Diagnosis is a big one, right? So these are a couple of the ones that I came up with. And the first one's really around prevalence and how common the disease is in the general public. And this is an important consideration because the frequency of disease changes with your approach and how you're going to develop a therapy for the disease. It's not a one solution fits all type of thing. Now, there are labs around the world that specialize in estimating these, and these are academic labs estimating the incidence and prevalence for rare diseases. Now, one I could think of is at the Broad Institute. It's the O'Donnell Luria lab, and they have published quite a lot of papers around estimating rare diseases. And I think that's an important piece of data that you can get out of research. Another one, and this is maybe the most obvious one, is of course understanding the disease biology. So in some cases, we don't even, in the rare disease space, there's frequently an underlying genetic mutation to a disease, and a lot of the times we don't know the gene. And I think over the last 10 years, we've really seen that explode, and we've seen sort of a renaissance around really biological understanding, where we've learned to appreciate what the genes are that cause certain diseases. But then we also want to understand what the product of that gene does in the cell. and then how mutations of that gene affect the functionality of that product. And for example, I have up here on the slide, it's gain of function, loss of function. It really changes our drug discovery campaign if we understand that we need to actually inhibit a protein versus activate a protein in order to rescue function. I think next we would then look at cellular phenotypes. So we look in cellular models, and depending on what diseases is, if you're talking about Alagille syndrome, for example, we may be looking at liver cells. We want to look at how the overall function and metabolism of the liver cell is affected by the mutation. And that will help us then build disease models that we can study in the dish. And that's the foundation for our drug discovery efforts. So the more we know about this, the less we have to do up front in order to actually start our drug discovery campaign. And then the last point that I have here is really around variants. And these are genetic databases like ClinVar, where you record variants in the gene of interest. And it's important to include disease-causing as well as benign mutations. And it will really help us not just in the estimation of the incidence of the disease, but it will also help us understand exactly what's going wrong with the protein that's encoded in the gene. All right, and then the last point I wanted to make here is really around diagnosis and the diagnostic journey. And I know that this is something that most of you probably know more about than I do, but I heard plenty of gruesome stories around diagnostic journeys and years where people have tried to get a diagnosis. And you probably all know that in order to be on the recommended uniform screening panel, the RUSP, you kind of have to have a therapy that's already commercialized or available. But I want to point out that there are disease-specific panels that are often sponsored by the industry. And one example of that is a cholestasis panel that we at Travere offer. And we've grown that cholestasis panel over the last, I'm trying to think, maybe 6 or 7 years. And it now includes over 70 genes that are known to cause cholestatic liver disease. Another example is a cerebral palsy panel. And I know that ADCY5 uses that, and that's provided for free with the help of PTC Therapeutics. So those are out there, and they're constantly growing. And I think if you feel like you're representing a disease that may fit in one of those panels, I would encourage you to talk to the people at the company, to contact that company, to maybe talk to some physicians, understand how it's being used, but also advocate to get the gene of interest onto one of those panels. Again, they're usually offered for free, and that we've noticed that really changes the diagnostic journey, and it's really helpful for physicians to who often struggle to get genetic testing approved by payers. And maybe in general, I just want to emphasize that companies are looking for partners as well, and they're often willing to help in any way they can. For us, the patient voice carries a lot of weight, and like I said, we've heard many gruesome stories around diagnosis and how long it's taken to get a real diagnosis, and we really want to change that. So the question is, how do you get started? I mean, how do you build your research network? What are some of the resources you can go to that will help you identify the researchers that are working on your disease or in the periphery of the disease? And some of this is probably very obvious and you're probably familiar with, but of course the first one I would start with is PubMed. PubMed is just an access portal to publications. There are millions of publications on PubMed, and you can search for your specific disease, for the gene that's mutated. You can look at many different paradigms or many different search criteria that are associated with your disease, and that will help you identify researchers around the world that are working in the space and that you could connect with. I think I don't have this on here, but also if you have existing contacts with researchers, ask for their suggestions. I think a lot of the time the communities are relatively small and people know each other. So feel free to ask for advice of who to contact, talk to. The second one is really, well, second and the third is really around funding sources. And I've listed here the report days from the NIH. That's all the funding that the NIH has provided. And you can access the projects. So you look for keywords, you can find essentially any funding that was given to a laboratory working on something related. The same exists for other countries. And here I have the German version of the NIH, since I'm German, the DFG. And you can find these sites, and it's a good way of going about finding who's working in a specific disease space. The third one is conferences, and with that, I don't necessarily mean that you need to attend conferences. I think sometimes it's, for your purposes, it's okay to just look at the conference agenda and reach out to scientists that are presenting on topics that are of interest to you and your community. I think one thing that you can always count on is that scientists love talking about their science. So don't hesitate reaching out to people. And then there are specialized platforms. I can think of one that's Altum, for example, and they kind of combine all these resources in one. And you can work through them and get a sort of a comprehensive list, a more comprehensive list, and you don't have to go through individual platforms. Now, if you have the bandwidth and your organization is set up for it, you can organize a symposium. Now, I think a symposium is important because it actually brings all the stakeholders together, and that is, physicians, patients, caretakers, and then industry. And we've had some very positive experiences in symposia, and we certainly make it a priority to attend patient-centric symposia. And then you can have scientific days, and you can have patient-focused days, and I think that usually works really well. I mentioned here the research hubs. As I evaluate disease spaces, I like to see research hubs for a very practical reason, and that is that it takes a very long time to embark on a collaboration with different enter-entities. We have to get a CDA in place, we have to get contracts in place, and that sometimes takes months and months. So if we have one preferred partner that has a lot of different areas covered, then that's our preference, and it really does work wonders, and we look upon it favorably when we evaluate disease spaces. And the final one is really to demand to be involved, and I think that's I have a little anecdote here, which is around our Alagille syndrome program, and the former president of the Alliance actually flagged down the head of NCATS, that's the drug discovery branch of the NIH that I mentioned earlier, and our former CEO at a meeting, and basically said, I want to collaborate with you. And at the time, we already had the NGLY1 collaboration, and so she just said, I want to have the same thing that these guys have. And she made the case. We did our homework, and here we are a few years later, and we have this program, and we're making great progress, and we're all equal partners in this endeavor. Okay, so this may be a good time to just see whether there's any questions about this first part or any additional comments.
If you have any comments, you can add it on the chat, and then we'll make sure that we read it to Karsten as well.
Okay, if there's no comments, I'm going to continue. I don't know how much time. When do I have to be done, Pavati? Is it 1:45, 10 to 1:45?
Yeah.
Okay. Well, I think we're on track. All right. So then let's talk about registries in natural history, and I think a registry in its minimal version is a list of patients with contact information. At the other end of the spectrum, a registry is a data collection with analysis tools and maybe even an associated biobank. So there's really a large range when it gets to registries, and accordingly, they can fulfill different purposes. I think a key word when I think of a registry is exploration. And the registry is really a tool to enable novel insights and new lines of research. And a couple of items that are here in terms of objectives fall into this category. For example, monitoring patient care and outcomes, advancing research hypotheses, observe patient behavior patterns. So all of those would fall under novel insights and new lines of research. But I think there's additional benefits to a registries, such as the ability to recruit patients for clinical trials more rapidly and to match the right patient to the right treatment trial. Additionally, patient registries can help with discussions with payers. Now, most groups don't have the resources to collect patient data properly or appropriately. So there are several non-profit organizations that provide platforms to help with that exactly. And the first one I want to talk about is IAMRARET, which is a little less than 10 years old. And this is from NORD, the National Organization for Rare Diseases. There was a program that was developed with the input of FDA, NIH, and KOLs. And I think the emphasis here really is on rare disease and the special needs of rare disease. Now, you're obviously all familiar with the challenges, but let me talk to you just briefly about how they relate to registries. So small patient population, right? I mean, it's hard to identify the patients. They're spread throughout the whole country. We talked about diagnostic delay. Obviously, there's physicians that don't actually know all the rare diseases out there, which are more than 7,000. And so if you have them in the registry, it helps not just put people in touch with each other, learn from each other, but also helps researchers reach out, industry to recruit, et cetera, et cetera. Hard to diagnose, delayed diagnosis. Well, I just mentioned that. The natural history, we're going to talk about this a little bit later. There's two different things. I'm also going to mention the distinction between my version of the distinction between the natural history and a registry. And then exploration, exploring endpoints, etc. Okay, so IAMRARET, there's probably about 40 registries for different diseases on this platform at this stage. This platform doesn't just offer you a home for your data, but it also offers you everything you need to get all the pieces in place, such as training, so guidance, documents, training documents, templates, etc., etc. Now, RareX is a recently launched initiative that's really meant to bring together advocacy groups, researchers, and industry. It's complementary in the sense that its focus here is very much on standardization and the structure of the data. It's really, the data is there, but how can you make it most useful? It is powered by technology out of the Broad Institute, which is Harvard and MIT, and it boasts certain data analysis features as well. Want to mention one other registry, that's the Sanford Health Registry. This is the oldest that I'm aware of. I think it was founded in 2010. There's over 80 different advocate or disease groups that have registries at Sanford Research. And then if you're interested in looking for registries, the most comprehensive tool that I could find to identify registries worldwide is this NIH tool, and maybe I'll give you a minute if you want to take down this this address. Now, when I'm asked to give advice to an advocacy group or a patient community about how to get to a registry, I really think that the two pieces of advice I always like to give is, have one big consolidated registry. Don't spread it out over multi different platforms. And the other one is really do your diligence and see which one is the best fit, which of these platforms is the best fit for your organization. They do offer slightly different tools and benefits. All right, let's move on to natural history. So registry data is often populated by patients and caretakers, and the aim is really to gather as much information as possible on a disease state. Now, in contrast, natural history studies are often formal clinical trials and are generally conducted more rigorously and systematically than registries. Rather than gathering as much data as possible, natural history studies are designed with specific questions in mind. So we're talking about questions around the outcome, rate of progression, life expectancy, or disease complications. And the ultimate goal is really to create a great understanding of how the condition develops and ideally how it can be most effectively treated. This is very useful in the rare disease space, and a couple of reasons down here. One of them is that there's substantial genotypic and phenotypic heterogeneity in rare disease, and that makes it very difficult to identify patient populations. What I mean with that, so a genotypic heterogeneity is where a disease state may not have just a single mutation or a single gene that's affected as the underlying cause. And a phenotypic heterogeneity is really where a mutation in a single gene may express itself in different ways in different patients. So it may affect different systems, organ systems. And of course, that makes it relatively challenging to identify patients. And I think natural history studies can really help with that. So can registries, by the way. The other one is really identifying, developing clinical outcome assessments. In a rare disease, there's often no precedent. And so how do we know that we're actually improving a patient? And I think natural history studies really help us with that. And we can look at ways to look at progression of disease, at remission of disease, et cetera, et cetera. The third one here is developing biomarkers. Now, biomarkers are generally molecules that are physiologically produced in the body and that correlate with progression or remission of disease. They're not necessarily looking directly at the disease processes, but they're representative of what's going on. And so ideally, robustly developed biomarker can actually look at the therapeutic effects of a therapy, or beneficial effects of a therapy. Okay, now I wanted to, yes, that's right. I wanted to give you an example where biomarkers can actually be used as surrogate endpoints under sort of an accelerated approval by the FDA. And we have a program that's similar to that. It's proteinuria, where we're looking for accelerated approval. And it's in kidney disease, and the biomarker is proteinuria. And the reason proteinuria is protein that's in the urine that's not filtered out by the kidney, and so it's a reflection of a compromised kidney function ultimately. But the real endpoints that we should be looking at is end-stage renal disease or kidney transplants, but that would take years and years and years of running trials in order to actually have enough patients reach those stages. And so the third surrogate biomarker here is proteinuria, or we can look at glomerular filtration rate, which is also functional readout of the kidney. And we have to establish that those correlate with the other endpoints I mentioned, like end-stage renal disease. And then the last point here, and that's a really important one for rare disease, especially for rare diseases where there's very few patients and that progress very rapidly. Natural history studies can serve as non-concurrent comparator arms in trials. So ultimately, some treatments are actually compared to the natural history because it's non-ethical to not treat patients in a clinical trial if they progress towards loss of function or maybe even towards death in the course of the trial. All right, I want to contrast two different forms of trials. One of them is the retrospective trial, and that's looking backwards, versus the prospective trial looking forward. Now, the retrospective trial is really where we use existing data, and we try to look at existing data to generate a natural history study from that existing data. Now, you can imagine that that's generally not as effective, because there's different ways of gathering data and reporting on it. And so the prospective trial brings the advantage that you can really set the format for how different aspects to the disease or the treatment are being reported. And so you can have consistency between different sites where you enroll patients. All right. Let's move on to the next slide and the next topic, which is around biobanks. And biobanks, they range as well. So in its minimal form, a biobank may have some blood samples for certain disease states. And there are global biobanks that have a lot of samples, but they have them from so many different diseases that there may be only a handful for each individual disease. On its other extreme end, you have very patient or disease-focused biobanks, and they include other cell types as well. So you may have biopsy samples, you may have induced pluripotent stem cells, which are cells that you can then differentiate into all kinds of cells that are found in the full organism, a human organism, and they may have sibling cells, so cells from non-affected family members for comparators. Now, I really tried to find an article that would demonstrate the impact that biobanks have on research, and I couldn't quite find a good figure for it, but this is an article that advocates the use of biobanks and tries to quantify how it improves or how it encourages research in a certain direction. Now, this is data from 1991 to 2016. I actually think that the data from the last 5 years would be even more insightful. And registries with biobanks, there's very little data on that. That's why they're so low. But I think the take-home message ultimately from this paper, that's somewhat illustrated in this figure, although not optimally, is that disease areas that have biobanks enjoy more attention from the scientific community and end up seeing a lot more publications. Now, maybe the largest biobank, but or at least one of the largest biobanks is Coriell. So you can see you can have access to many different repositories that are associated with a list of different institutes. You can also deposit cells here, but because these biobanks are covering many different disease states, there may be an upper limit to how many cells you can offer. You can deposit. Coriell offers more than just a spot in the freezer. They also offer sample kits, sample collection kits, DNA analysis, and sequencing. Two other biobanks that I want to mention, but they are just representative, which is for medical centers. So there's a lot of medical centers that have their own biobanks. And the two that I want to mention here is, again, at Stanford Health and then at Children's Hospital of Philadelphia. And some of these are actually more disease specific. And so there are a long list of samples for a specific disease state. I did want to mention that specific disease-specific state, specific disease state. One example that I could think of was Friedreich's ataxia biobank that is at the University of Alabama, and so you can access a lot more samples if you have sort of a disease-specific biobank. And then I'm aware of this tool. This is from sort of a former life when I was still living in Europe. Where you actually have a Euro biobank, which gives access to 25 different biobanks in 11 different countries, and so you can basically, rather than having to screen through each individual one, you can just look for something in that state on this platform, and see whether it already exists. Maybe for a researcher perspective, you can find the cells that you're looking for because you have access to a whole lot of different resources there. All right, then Biobank Finders. These are two platforms that, unfortunately, they're also focused on Europe. I couldn't find the equivalent for the US, but these are two platforms where you literally just enter the disease state that you're interested in, and will search for biobanks that have samples with that disease state. Now, most of that is Europe. There's a little bit of Asia in OrphanNet. RD Connect, I tried to get on them. I used to use them, but I tried to get on them earlier this week, and I couldn't. So I don't know whether it still exists. I do know OrphanNet exists, and I do know it fulfills all the functionality that RD Connect did as well. All right, so I've covered the first three topics. I think we're right at Q2. So I won't talk about clinical trial jargon right now, but feel free to reach out to me if you want to learn a little bit more about that. Happy to share my email address with you as well. And I can do my best to share materials that will help you understand some of the jargon that we use and clinicians use, and that is relevant to rare disease drug discovery. All right, I think we're ready to go to Q&A.
Hi all, you can either put your questions in the chat and then we can ask it in the order that they come, or if you can raise your hand, then we'll just call on you so we can make sure that everybody gets a chance. So yeah, if you have any questions, please feel free to let us know. Some of the questions, Karsten, that we did get, I'll start with those, if that's okay.
Sure.
What are the top 3 priorities to be research ready, and some activities that people might think are important but actually aren't?
That's a really good question. I mean, so I think one thing that I didn't talk about, it's good to have a scientific representative on your patient advocacy group, on your board. Have somebody out of academia, maybe even out of industry, who understands the disease space from a science perspective, who can represent you in conversations, who can, he can keep track of the science and the new developments. So I would think that trying to recruit somebody to your board is probably one of the first priorities. And then I think after that is really what I put in the first place here is growing your research community. I think the better your network, the more cross-fertilization will occur. There's going to be more collaboration, and I think it's not so different being a rare disease researcher than to being a rare disease patient, in the sense that you're somewhat isolated. There may be nobody else within 100 miles from you working on the same disease state, and so I think being able to make those connections is valuable to anybody working in the rare disease space. So that's a tool.
We have a question from the audience here. Which of the 4 themes is Travere Therapeutics looking to improve to be more research ready? Is there a particular rare disease that Travere is trying to address in the coming months?
Well, I can't really talk about non-public data. I mean, we obviously have these 2 discovery programs in NGLA1 deficiency and Alagille syndrome alliance, and we're committed to those. We are working with the right partners in that space, and so we do everything we can to try to get therapies into development, and then ultimately, of course, into clinical trials for those 2 indications. And outside of that, I can't unfortunately discuss sort of what our thinking is.
This is a great question. When you mentioned some clinical endpoints, maybe too long-term to fit a study, can you explain a bit more of what can get used as a proxy for these?
Yeah, that's a really good question, actually, and I wouldn't say it's a moving target. I just think it's challenging because ultimately they're not. There are examples out there for it, but they're not that many, and I think all of them have come more recently where there's no where it's a case-by-case scenario, right? I mean, I think the example that I mentioned with end-stage renal disease or kidney transplant, and then sort of a biomarker for kidney function is a good one. And I think those would the similar examples would apply in other disease states. There's imaging biomarkers. So if you get MRI imaging, for example, for you can get ultrasound imaging biomarkers, et cetera, et cetera. There's density scans for bone diseases, et cetera, et cetera. So I think those are ultimately those would all be considered surrogate biomarkers. Sorry, does it answer your question? I'm not sure whether I did a good job of answering that question.
I think so, and if not, Daniel, please ask us.
Yeah, please follow up on it if I didn't answer it.
How do you deal with physicians in your disease that can only focus on treating newly diagnosed instead of working with those who have had, who have been diagnosed for a while with the disease?
Okay, can you read that question again? I just want to make sure I understand it correctly.
How do you work with physicians in your disease state that are only focusing on newly diagnosed patients versus working with those who have had the disease for many years?
Yeah, I'm trying to understand that question. I think there's what I can think of is that there's a lot of pediatric doctors that don't follow patients once they reach adulthood. And I know that from the communities that we work with, that some of those patients really have a hard time transitioning to transitioning their care into adult care, and I know that that's a challenge for them. I'm not 100% sure that I'm the right person to answer that question, but I've heard of examples where even patients into their 20s have been continued to be seen by their pediatric doctor because he or she is so familiar with the case and with the treatment paradigms that are out there. Again, I'm not sure that I'm the best person to answer that, but maybe that'll help a little bit.
Pros and cons of the different modalities that exist today.
That's a very good question. So I didn't get into that, but of course we could spend a whole hour or several hours discussing that. I think the great pro is that we're developing new modalities that help address more and more diseases that are out there, and ultimately address them more satisfactorily. If we can replace a gene that is mutated using gene therapy, we can essentially reverse the disease. And we can reinstate a completely healthy individual. I think that's that there are when we weigh up different modalities, it depends what organ we're trying to target. It also depends on ultimately what are we having to do, right? I mean, for example, we talk about antisense, so knockdown of genes, right? If we have to activate a target protein, we can't use antisense therapies. On the other hand, it's hard, not impossible, but it's hard to inhibit a protein using gene therapy. Again, you would replace that. So it depends on what you're trying to achieve and where you're trying to achieve it.
This is from one of our international members. What limits drug development in developing countries?
So let me try to. There's a lot going through my head right now. I'm trying to think of whether that question may come from a perspective of clinical trials in developing countries. And I think part of that is simply infrastructure, right? I think there are certain countries that I guess most companies prefer to work in for clinical development. And I think with developing countries, the issue a lot of the time is just having the infrastructure available to have some of those readouts that we're talking about. Let's talk about an MRI surrogate endpoint. Obviously there has to be sufficient access to the right tools and machinery in order to screen that.
The next question is, how has government played a factor in the development of research-ready networks from your experience?
So research-ready networks. So I'm trying to think of what you're referring to here. Maybe networks between different scientists or different physicians. I'm probably on the academic side most familiar with the European model, since I did most of my academic training in Europe, and there are centers of excellence in Germany, for example, for different types of biology. Those are funded by the government, and they get they're all funded by the government, but they get extra funds to be a center of excellence. And similarly, we have centers of excellence in Switzerland, where I did my PhD. In the US, I apologize, but I'm not quite as familiar with that because I've pretty much done all my industry work in the US, and so I haven't been exposed to that as much.
Another question: what type of information do researchers and industry want, and in what format does it work best?
What type of information
are they looking for from patient advocacy organizations, and in which format would it be best?
Beyond, yeah. That's a very good question, because it can take many different forms and it could really be interpreted in many different ways. But if we're talking about sort of a disease understanding, I kind of touched on that a little bit. Obviously, we would prefer if we could start with the drug discovery right away. Basic research takes a long time. It's very arduous, and there's a lot of setbacks. So if we have a good understanding of the disease mechanisms, I think we feel like we're enabled to actually try to target it. And I'll give you the example of NGLI1 deficiency that we're working on, and where we do need to do some basic biology, because it's a very small disease state, and there's not a lot of researchers working on it, and we don't fully know what it does in the cell when you're missing NGLI1, and that is a very very tough hurdle to overcome. We have to spend a lot of resources trying to really understand the science behind the disease better. In terms of I guess in terms of really sort of the profile of an organization, and maybe that's what you're referring to, I think we're not necessarily as interested in funding. I realize that there's a lot of organizations that are well funded, and then maybe there's a few organizations that are well funded, a lot of them that are not, but that's not really a key to us. I think what we feel like we need to have is an organization that can reach out to patients, that has access to patients, that has a voice in the patient community, that represents the patients in sort of on their behalf, and that tries to think of different ways of helping patients. We've had to learn that it's not just drug discovery that we can contribute. I guess I shouldn't say we've had to learn. We've enjoyed learning that we can help in many different ways, not just with drug discovery. And I feel like patient advocacy groups are in the same boat. Sorry if I'm preaching to the choir here, but I feel like that's something key to us. We feel like we want to work with organizations that have the same philosophy as we do, in that sense.
And this is our last question from what I gather. How can we get more young graduates interested in rare disease research? And this particular question, it was about sickle cell.
Yeah. Yeah, that's a very good question. That's one that I've discussed with the Alagille Syndrome Alliance quite a lot. Obviously there's sort of grants that you can give to young trainees. I think that has some effect, maybe not as much as you would hope. I do think that having industry interest in the disease does change how it's being viewed by trainees and by people deciding on their career and their research focus. I was not necessarily interested in transitioning in industry when I started my academic career. I was fortunate in the sense that I encountered a company that was working exactly in the same space as I was doing my academic research. But I think there's a lot of people that think long term. They think about what chances do I have to get a grant money from the NIH or to find an industry job with that expertise. And I do think that that's changing as we see more interest from industry to develop treatments in rare disease specifically.
Okay, we made a last call for questions, but we're also at the top of the hour. So thank you all so much for taking time to attend this meeting, and a huge thanks to Karsten for joining us, answering all our questions, and for taking time out of your busy schedule to be here. Thank you all once again. We hope to see you June 8th through 10th in Philadelphia as a part of our main event, the Rare Drug Development Symposium.
Thanks for having me.
Thank you.